just make sure that we're broadcasting.
I don't know how much of my notes I really need for the honor of introducing my colleague and good friend, Dr. Barbara Clark, who's an associate professor of biochemistry and molecular genetics. Barbara's a graduate of Miami University of Ohio, and then did her Ph.D. at UT Southwestern, where she worked with Michael Waterman, and published 5 papers on the structure and function of cytochrome P450 hydroxylase enzyme. She subsequently moved to Texas Tech to work in Doug Stocko's lab. And there she cloned and named the steroidogenic acute regulatory protein STAR, which now is the first and founding member of the STAR-D family that she is going to talk about today, STAR-D5. She was recruited to UofL in 1995 and was tenured in 2001. Most recently, she served as acting associate dean for the graduate school and has now returned to the department full-time. She's established a productive research career with 77 peer-reviewed research papers, 14 peer reviews, and 18 book chapters. And she has a number of collaborators, and I have benefited greatly as a collaborator from her wisdom and kindness over the years. She's trained a number of graduate students and postdocs and technicians who have gone on to good careers, and she's currently serving on a number of Ph.D. committees helping to identify key questions in current research problems that really aid students. In addition to her research, Barbara has been active in graduate and medical education, teaching in the first and second year medical curricula, as well as serving as course directors for graduate courses in molecular biology, molecular toxicology, and the BMG literature review class. Let's see. In addition, she has been involved in a variety of service activities at the institutional, school, and department level that are too many for me to ask. In addition, she has national service on a variety of study sections, including as chair of American Heart Association panels, and has served on DOD and NIH panels. She is currently the deputy editor of the Journal of Endocrinology and the Journal of Molecular Endocrinology, and she's a PI on a pilot project grant from the CIEHS P30 Center here, and I think that's some of the research that she's going to discuss with us today. So Barbara, thank you for talking with us today.
Well, thank you, Carrie, for that kind introduction, and it's my pleasure to be here today to talk to you about one of my favorite proteins and topics. And as the title says, we'll be focusing on STAR-D5, which is an intracellular cholesterol transporter protein. And we'll look at potential functions for STAR-D5 and cholesterol-associated steatohepatitis, or CASH. So this is full disclosure, nothing to disclose. And the talk will follow this outline. We'll look first really at NASH, NAFLD and NASH, the liver disease, which this group I'm sure is well familiar with. And what I want to focus on, though, is this cholesterol-associated steatohepatitis, because this is something that is new, at least in my reading, and I think there's a group that is promoting this cholesterol-driven disease progression to inflammation in a subset of patients that this could be happening. And then we'll end looking at the recent data that I have from the research on using the STAR-D5 knockout mice and how that may contribute to disease progression. So to begin with, to get us all oriented on the same page, what the underlying interest of my lab is understanding lipid metabolism dysregulations and potential contributions of the START proteins to that, and the non-alcoholic fatty liver disease, or NAFLD. And so what we see here is that let me get my, As we know that you have accumulations of lipids within the liver, that results in fatty liver disease, and what's shown here in these white spots is where lipid droplets have accumulated in the liver, and you typically do not want to see this. So that is non-alcoholic fatty liver disease, meaning alcohol is not driving this disorder. But as you have increased or under chronic conditions, and what we'll be looking at, endoplasmic reticulum stress or ER stress, as a fundamental underlying driver of progression of disease from simple steatosis or NAFLD to a state that has inflammation and fibrosis, which is NASH, non-alcoholic steatohepatitis. And NAFLD and NASH are increasing in incidence, as we know, because of the increase in obesity and diabetes, type 2 diabetes as occurring, with insulin resistance, which is underlying both of those metabolic disorders, being a driving factor for this dysregulation in lipid metabolism, contributing to NAFLD and NASH. But typically, when you move from simple fatty liver disease onto more inflammation and fibrosis, it requires what is called a second hit or a third hit or maybe a multi-hit that is required to have disease progression. And the second hit after insulin resistance could be genetics, or here at UofL there's this very strong interest in environmental impact on health and disease. So if an environment or a chemical could be a second hit in combination with genetics and insulin resistance, this has been termed toxicant-associated steatohepatitis by Dr. Matt Cave and from his group of work. So what I want to look at next is this CASH, or cholesterol-associated steatohepatitis, which is another acronym for disease. And I will say at the end, it is my understanding that the patients there will be presenting with NASH, the inflammation and fibrosis that could proceed on to cirrhosis. It's just what is the hit? And if there are multiple hits, and can we identify multiple targets that we could potentially treat patients that have more of a directed approach. For some patients that might be whatever that second hit, whether it's genetics or environment, that we can potentially combat the disease progression. So when we think about non-alcoholic fatty liver disease, this comes from looking at the liver lipid panel profile from control cases, simple steatosis, and steatohepatitis patients. Now, this is only N and a 9 for each. But typically, when you think of fatty liver, an accumulation of lipids in the liver, we consider fatty acids and triacylglycerol accumulation. And you can see here, this is what is increasing. Here we have the short chain monounsaturated and polyunsaturated fatty acids are increasing. And if we look at the triacylglycerol content, we see a marked increase as we progress from NAFLD, from normal to NASH disease. And what I've highlighted here is this free cholesterol. You can see that free cholesterol is also increasing in these patients. And in this CASH hypothesis, it's really this free cholesterol levels that they are studying as potential driver for disease progression from NAFLD to NASH. And if you look over here, the ratio of free cholesterol to the phosphatidylcholine, a phospholipid, we look again at from the normal liver to NAFLD and to NASH, we see that in these patients, there is detectable levels of increase in free cholesterol. So what is the cholesterol-associated steatohepatitis model? Well, this comes out of George Iannawu, if I'm sure I'm not pronouncing that correctly, from University of Washington. And this is a review in the recent hepatology communications. And what you can see, what is depicted here is, again, what can lead to increase in that hepatic cholesterol levels is lifestyle, diet, obesity, as well as genetics. But what is depicted here is that it is this central to the development of steatohepatitis is this free hepatic cholesterol. And that in combination with the second or multi-hit, whether it's a gut microbiota, which is another big topic, insulin resistance, diabetes, adipokines, that will promote disease progression onto cholesterol-associated steatohepatitis. And again, just a quote from this paper is that in the subset of patients, they're proposing that it is this accumulation of free cholesterol that results in the development of this type of disease, CASH, and that it is actually the main driver of the inflammation and fibrosis. So if we think about cholesterol handling in the liver, this is, again, just to look at how cholesterol uptake and synthesis occurs in the hepatocyte in this case. We know we get cholesterol from circulating lipoproteins, either the low-density lipoprotein through receptor-mediated endocytosis, or after a meal through the chylomicron remnant through its own receptor. And basically through processing through the endosome and lysosome, you have free cholesterol levels that can be converted to cholesterol esters through the action of the enzyme acylcholesterol acyltransferase 2, or ACAT2. Second source of hepatic cholesterol is through de novo synthesis, and this is shown from acetate to this free cholesterol pool, and the rate-limiting enzyme in that pathway is HMG-CoA reductase. Also what's shown here. There a question from online? Oh, I'm hearing myself. Okay, that's not a good thing. It's Halloween. No. So you can see that we have other transporters that can contribute to the pool, as well as uptake from circulating HDL. Now, this free cholesterol levels are maintained, and basically free cholesterol is either stored as cholesterol esters and can be packaged into very low density lipoprotein, or VLDL, for delivery back to the adipose tissue where it should be stored in the lipids there. So once these free cholesterol pools are increasing, again, you can, as I said, through VLDL, take that back to the adipose tissue, or you have free cholesterol going into the bile through the ATP-dependent transporters, ABC, G5, and 8, or through the bile salt export pump. You can have free cholesterol converted to bile acids and then going into the bile. And these are a couple of the enzymes that regulate that pathway. And also, cholesterol then is removed from the cell through the ABC transporter, ABCA1, where it is loaded into HDL for a reverse cholesterol transport. So this just is to show you that there are multiple enzymes in this pathway that can be controlled to regulate the level of free cholesterol or cholesterol esters that are in the liver. And this is a highly regulated process where the level of free cholesterol will control its homeostasis, so you don't have buildup of free cholesterol within the liver. And part of that regulation is through a transcription factor. This is steroid regulatory element binding protein 2, or SREBP2. It's a sensor of cholesterol levels in the endoplasmic reticulum. And when those levels are low, we have high levels of this transcription factor in the nucleus that will increase the expression of the rate-limiting enzyme in cholesterol biosynthesis. So here, again, it's showing that biosynthesis and diets will contribute to the free cholesterol pool in the liver. And when those levels get high enough, this process will be inhibited. They'll feedback and inhibit this mechanism. The problem is in NAFLD and in NASH, this is dysregulated. So in the presence of high cholesterol levels in the liver, we don't turn off the transcriptional programming that leads to increased de novo synthesis. So in the presence of high cholesterol, we still observe higher levels of the transcription factor SREBB2, increase in HMG-CoA reductase, and so cholesterol synthesis continues even though the levels are sufficiently high, and that process should be turned off. In addition, what's typically observed in NAFLD is that you have an increase in the cholesterol ester hydrolase that will release free cholesterol from the cholesterol esters, as well as a decrease in these transporters that will remove free cholesterol from the liver. So this all combined together, this dysregulation, will promote that increase in free cholesterol in a NAFLD and NASH conditions. And so when this happens, the liver tries to protect itself by inserting more of the cholesterol ester into lipid droplets as a potentially inert, because free cholesterol in membranes, as we'll see, is not a good thing if it's overexpressed. So here, what's shown here is a model or cartoon of a lipid droplet or a cholesterol lipid droplet, and here you have the phospholipids, and cholesterol is usually in there with maintaining the phospholipid monolayer for the lipid droplet. Inside here, we see this cartoon here, the cholesterol esters and triacylglycerols, and that's fine. The problem happens when there's too much cholesterol, and it basically it has to pack upon itself. It's a very hydrophobic molecule, so it starts forming what's called cholesterol crystals at the edges of these lipid droplets, and it's these cholesterol crystals that are recognized by some of the cell types in the liver to try to get rid of those, and that promotes some of this disease progression. So how do the cholesterol crystals in these lipid droplets promote hepatic inflammation? Well, here you see if you have this lipid droplet in hepatocyte, these crystals promote this NLRP3. This is a pro-apoptotic or pyroptosis, which is a form of cell death. So you start getting a necrotic hepatocyte. This will result in the recruitment of the Kupffer cells, and the Kupffer cells then will uptake more of these cholesterol crystals and activated foam cells, basically by recruiting more of the macrophages around to the inflamed liver. And with the recruitment of these immune cells, there will be secreting for the pro-inflammatory cytokines, and this can lead to activation in the hepatic stellate cells, which will further secrete factors that will promote fibrosis. So altogether, that's one of the promoting factors when you have the appearance of these cholesterol crystals in the lipid droplets in the liver can activate the Kupffer cells, macrophages, hepatic stellate cells, which is what we observe when we're progressing to NASH. So cholesterol, though, is important. We need cholesterol in our membranes, and just like cholesterol homeostasis is well maintained, the distribution in our cellular membranes is regulated and maintained. And so we have higher levels in the plasma membrane, lower levels in the ER, and there are transport proteins within the cells that maintain this distribution of cholesterol between the membranes. And shown here again is just a consequence of what happens if you overload either the plasma membrane, the endoplasmic reticulum, or the mitochondria membrane. All of this can lead to disruption in metabolism. So if we look, I just went through this overloading of cholesterol in the lipid droplets can lead to Kupffer cell activation and fibrosis. But if we still have overloaded cholesterol in the ER, this can lead to inflammation and this cell death. You have overloading cholesterol in the mitochondria and the outer membrane that can activate ROS signaling, releases cytochrome C that promotes apoptosis. And in the plasma membrane, there's studies showing that it activates signal transduction pathways. This is a transcription factor, TAZ, that is involved in inflammation. So this basically is a hedgehog signaling pathway, and this factor that is activated works on hepatic stellate cells for activation of inflammation and fibrosis. So consequently, if you have elevated free cholesterol, whether it's in the crystal and the lipid droplets, whether it's more in the mitochondria or in the ER or the plasma membrane, can promote ultimately inflammation and fibrosis in the liver through multiple pathways. So it's important that we maintain cholesterol in the right spot and in the right proper distribution. So that brings me to the steroidogenic acute regulatory protein family. So this is the star domain protein family. And what I've shown here is just a structure modeled from the crystal structures of the start domain, and indicated here, there's a very long hydrophobic groove that binds sterols and lipids. In the mammals, there are 15 known members, and all of these members bind lipid binding proteins. And we are going to focus on 2 members that bind cholesterol. And this is a model of the non-vesicular sterile trafficking by the start domain protein families. And again, there's more than just, we can see that here at the mitochondria, that we have a star. This is what Dr. Kling introduced. This is what we first purified, identified, and showed that is critical for movement of cholesterol into mitochondria. And this is mainly important in steroidogenic cells for production of steroid hormones. However, there's a recent report that came out that some hepatocyte-specific star can drive an increase in bile acid synthesis in the liver, which can promote some toxicity in the liver. But we'll focus here now on these 2 transporters, STAR-D4 and STAR-D5. And today's talk is going to be focused mainly on STAR-D5. These are 2 what we call soluble transport proteins. This is the whole structure of both of those proteins are very similar. They both bind cholesterol. They both bind oxysterol. There's nothing in the protein sequence to give us any indication of what membrane they should be associated with. So the question is, what are they doing to move cholesterol between the membranes within a cell? And from in vitro and in vivo studies, it has been shown that STAR-D4 likely is involved in cholesterol recycling between the early endosome and the plasma membrane, as well as bringing cholesterol from the plasma membrane to the endoplasmic reticulum. And there it activates this ACAT enzyme to promote cholesterol ester formation and lipid droplet formation. STAR-D5, on the other hand, has been shown to enhance the flux of cholesterol from the ER to the plasma membrane for efflux to the circulating HDL. So we can think of STAR-D4 mechanism as being important in keeping cholesterol within the cholesterol ester form within lipid droplets, and STAR-D5 as being important for removing cholesterol out of the cell back to HDL, or moving it from the ER to the plasma membrane. Now, I make those statements like this is, solidly set. But we still, these are models that we are using to design experiments to fully test what the function of these soluble cholesterol transport proteins do. Over a decade ago, a mouse, STAR-D4 knockout mouse was generated, and there's no phenotypic effect on that. And one possibility, or likely possibility, is that there could be compensatory mechanisms occurring in the cell through these other start domain proteins or through other cholesterol transporter type of proteins that are going to make sure that we maintain cholesterol in the proper distribution within the cell. So up to this point, I've tried to convince you that if we have aberrant cholesterol trafficking within the cell and distribution within the membranes, that it could potentially lead to the disorder and that we observed in that NAFLD and or NASH. So we're going to focus now on STAR-D5 and ask the question, if we remove STAR-D5 from the liver, will that impact lipid metabolism, and ultimately maybe ask the question of cholesterol distribution. So we have generated a STAR-D5 global knockout mouse. This is done through CRISPR-Cas9 gene editing. And we have now that founder line was back crossed onto the C57 black 6J line, which is classically used for studies with NAFLD and NASH. And step 2 is to address this question is that we use tunicamycin. Tunicamycin is an ER stress inducer, and this is just as a proof of principle to see if there was any effect of loss of STAR-D5 on hepatic responses, specifically focusing on lipid metabolism. So we use 2 to 3 month old wild type and STAR-D5 knockout mice and treated them with the control vehicle, DMSO, or tunicamycin for 1 day for a short-term treatment, or tunicamycin for 4 days, which is a longer treatment. And we then selected specific target genes that we knew were important in regulating cholesterol and fatty acid metabolism in the liver, and did RT-qPCR analysis to look at those changes. And what we observed is that there was no genotype difference and we'll see this data in just a minute, no genotype difference between the knockout and the wild type mice without any inducer. And it took a chronic treatment with tunicamycin before we observed any differences. So now why this is just in here to also, I'm using tunicamycin even though I am interested in diet-induced and environmental-induced NAFLD and NASH. Using tunicamycin because we can see these results in 4 days and not 4 months, and to give us some preliminary data to support further pursuing STAR-D5. So if we look at ER stress and why that is related to NAFLD progression, this is just showing the ER stress proteins that are localized in the endoplasmic reticulum, ATF6A, IRE1 alpha, and PERK. Each of these are activated when there's disruption to the ER. So tunicamycin treatment will result in activation of all 3 of these pathways. And what the downstream responses, all of these will result in transcriptional reprogramming. And what is observed is with those that you can see that you get increase in insulin resistance, some increase in fatty acid transport to the liver, increase in free cholesterol, increase in VLDL production, and this is classically what is observed in NAFLD or steatosis. Now, if ER stress can resolve itself through transcriptional reprogramming, or if there's too much damage, it can go on to activate inflammatory, oxidative stress and cell death processes, and potentially even on to more severe phenotype or that onto hepatocellular carcinoma. So this is showing that as the disease progresses, the ER stress increases because lipotoxicity, this accumulation of lipids, can feed back and further promote the ER stress that occurs. So using tunicamycin, we thought that would be a good approach to see if there was any differences in our STAR-D5 knockout model or compared to the wild-type littermate controls. And so this is just summarized here again, the idea being that you have this ER stress, lipid metabolism dysregulation. We expect to see an increase in cholesterol. We know STAR-D5 functions to move cholesterol to the plasma membrane for efflux to HDL as a compensatory mechanism. But under conditions where there's high levels of cholesterol, is it possible that STAR-D5 might be promoting aberrant accumulation of cholesterol in the plasma membrane. And if you have elevated cholesterol in the plasma membrane, it is known to activate toll-like receptor signaling and to inhibit EGF receptor signaling. Now, both of these are known in the non-alcoholic fatty liver disease. The combination that the toll-like receptor signaling can increase proliferation, inflammation, and fibrosis, and EGF receptor signaling would usually suppress these activities, I would say. So if we have a loss of EGF receptor signaling and activation of toll-like receptor signaling, that could be one potential mechanism where we have aberrant plasma membrane cholesterol leading to this dysregulation observed in NAFLD and NASH. So that's our model. So now basically we're asking the question, with tunicamycin-induced stress and the absence of STAR-D5, do we see any difference in this lipid metabolism dysregulation? And some data now, we looked at treating our mice, the wild type and knockout, with tunicamycin, shown here in red. And you can see in the wild type mice, if we look at STAR-D5 expression, this is looking at the RNA level through RT-qPCR. We see that there is, relative expression here in the wild type mice. We have STAR-D5, modest but not significant effect on increasing the mRNA of STAR-D5, but we completely lose STAR-D5 expression in our knockout mouse model as we anticipated. But looking at an ER stress responsive protein, which is GRIP78, we see a robust induction of GRIP78 in the wild type and knockout model, indicating that tunicamycin has induced ER stress in both these animal models. Using Western blot analysis for STAR-D5, again, we can see that with tunicamycin treatment, we observed a small but non-significant increase in STAR protein expression. But most importantly, we have no detectable STAR-D5 protein in our knockout animals. And after tunicamycin treatment, we still don't observe any detectable STAR-D5. And over here with the GRIP78, it just confirms what we saw at the RNA level, that we get a robust increase in protein expression of GRIP78 in both the wild type and the knockout animal. So both of these animal models are responding to tunicamycin with an ER stress response. So just quickly, I'm going to focus only on a few of the target genes that we looked at that are involved in cholesterol pathway. So this transcription factors LXR and SREBPF2, as well as the HMG-CoA reductase, the rate-limiting enzyme in cholesterol biosynthesis, and ABCA1, which is the efflux transporter out of the cell. And this is a busy slide, but what we will look at here is each of these cases. In the black is a wild type. This is the DMSO control, 1-day and 4-day tunicamycin treatment. And then in the red are all the knockouts. And I'll bring your attention, it's hard to see if you just look at this first set, that when we look at these targets and compare the wild type DMSO to the knockout DMSO, there's no change in expression. So simply knocking out STAR-D5 has no basal effect on any of these target genes. With tunicamycin, I see the same thing after 1 day with tunicamycin treatment, that even if we have a potential decrease in some of these proteins, there's no genotype difference at the 1-day treatment. However, when we look at the 4-day tunicamycin treatment and compare the wild type to knockout, we begin to see a difference, such as shown here with ABCA1, where we see a decrease in the STAR-D5 knockout mouse compared to the wild type mouse. And looking over at this pathway analysis, some of the other genes that we looked at, when we looked at bile acid synthesis, we saw no change between the genotypes, but we did see a decrease in the bile acid converting enzyme. That was with tunicamycin treatment, but there's no genotype effect. Similarly with HMG-CoA reductase, we saw a tunicamycin effect, but we also saw that it was more pronounced in the STAR-D5 knockout. And so when you see these double arrows that we're showing that the tunicomycin effect that we observed in our wild-type mice and our knockout mice is consistent to what's in the literature for what happens in the liver, lipid metabolism with tunicomycin treatment. But we did observe a difference in the knockout animals compared to the wild-type animals, suggesting that they are handling lipid metabolism differently after the tunicomycin-induced ER stress. So with that, we went and funding through the CIEHS here to do an RNA-seq analysis on the RNA from the 4-day tunicomycin-induced liver transcriptome, comparing the knockout to the wild-type animals. And this would give us a much broader, unbiased approach because what I failed to mention, the STAR-D5 is expressed not only in hepatocytes, but also in Kupffer cells, macrophages, and potentially the hepatic stellate cells. So it's expressed in all cell types of the liver. So where it might be having an effect is a question that we want to answer. So in this case, again, we took 2 to 3 month old knockout mice and littermate control mice, treated them for 4 days with tunicomycin, and then did the RNA-seq analysis from the isolated liver. And again, this just is recognizing that the Center for Environmental Health Sciences was used, and the core is there to do the library prep and the bioinformatics. Here's the heat map. Then this is certainly not so that we can recognize any genes change, but to give us a very nice visual at first that with the DMSO, these whole groups of these 10 animals, they are randomly assigned whether they're knockout or wild type for each column. But if you compare, if you just look at the colors across here, you can see that globally there's no transcript changes between the knockout and the wild type in the DMSO treated samples. However, if you look at the TUNI treated samples, you can see that there are clear differences between the wild type and the knockout globally. So there's a lot of transcripts changing there. And some of this is one of the main differences in the knockout TUNI treated animals is the response to the ER stress is repressed or lower. So that is when we look at the differential gene expression, and again, this just what we saw visually, comparing the DMSO treated to the wild type animals. This is total number of genes, and then we kind of parse this data out by looking at only those genes that are significant and that are differentially regulated, have a false discovery rate less than 10%, and that they have a log fold change of 2, which means there's a 2-fold difference in expression that we detect. And when we compare just the untreated, we can see there's only 7 upregulated and 2 downregulated genes, one of those being STAR-D5, which was the first thing we looked for because that would not have been a good thing if we didn't see that. So when we look at the treatment here, looking at the wild type, the tunicomycin to the DMSO control or the knockout tunicomycin to the DMSO control, again, we can see the total number of genes that are changed in both the up or down regulated pattern. And then what I focus on, I'll show you some data on when we looked at the interaction. That is, if we compared the tunicomycin treated knockout animals to the tunicomycin treated wild type animals, we have a number of changes, almost equally up and down regulated genes in that packet. And if putting this gene list into a database analysis, Metacore database analysis, to understand pathways that might be altered between these due to the genotype, top pathway analysis that came out was for CAR, which is a nuclear receptor that is involved in drug metabolism in the liver. So this is xenobiotic metabolizing enzymes. Here, which was very reassuring and rewarding rewarding, yeah, I guess that's right, was looking at this SREBP pathway that controls cholesterol and fatty acid biosynthesis. Those were two of the targets that we had originally identified. Something I want to follow up on but haven't done yet is the retinol metabolism, because that occurs in the stellate cells, so that might be of some interest. And then again, sirtuin regulation. PXR is also a nuclear receptor involved in drug metabolism, as well as the role of ER stress in obesity and type 2 diabetes. So again, if we look at, I've just highlighted in red and green what's up and down in between the knockout and the wild type after tunicamycin treatment. You can see a lot of the same gene targets are being used to generate this pathway analysis. And so I took those common gene targets and made a pathway analysis to summarize the data with the focus on cholesterol metabolism and bile acid transport. Again, this is a little busy slide, but this is just showing the ER stress response genes. These are all known to be tunicamycin responsive. These are all up. This also indicates that tunicamycin was inducing ER stress. And we can see that all these genes are more highly expressed after tunicamycin treatment in the wild type animals compared to the knockout. The second level, these genes will then control this level of genes. And what we observed is that in the fatty acid synthesis, some of these target genes here, fatty acid synthase, acyl-CoA carboxylase, they are down, but there was no genotype difference. And this is consistent with the literature with what is observed with tunicamycin treatment. And we saw some of these targets that we see are down equally in the wild type and knockout, or down only in the knockout, in the wild type. These are cholesterol biosynthesis genes. I'm pointing this out because it's not all or nothing, is that the STAR-D5 knockout animal has differences in these key target gene expressions that are differentiated from the wild type animal. But it's complex, as you can see, because the wild type animals are responding in their own way, and the knockout animals are responding their own way. But overall, what we observed is that the changes in the cholesterol metabolism genes is more suppressed in the STAR-D5 knockout animals. As you can see, most of these with the wild type are indicated here. I will point out that STAR-D4 came up in our analysis, and this was, I will say, a surprise because this hadn't been in the literature. And so we picked a few of these target genes, one that had equal changes between the genotypes, one that was only down in the wild type, and one that was down in both, but a differential response between the two genotypes, and followed up with the Western blot analysis. And that's shown here with the STAR-D5. As we expected, we don't detect any protein in our knockout animals. With STAR-D4, we see tunicamycin ablates its expression in the wild type animals, but it's not so, it's still less responsive in the knockout animals to tunicamycin treatment. And similarly, we see the same with this enzyme within the cholesterol biosynthetic pathway. Tunicamycin ablates its expression in the wild type animals, and the response is suppressed in the knockout animals. While fatty acid synthesis has the same response in both animals. And again, we saw GRIP78 indicating that the tunicamycin response is intact. And this is just showing the log fold 2 change for the RNA for all these targets that I just went through and the Western blot analysis. And this is the quantified data for the protein expression. So we can detect differences at the RNA level and the protein level between some of these major regulators in cholesterol metabolism, bile acid metabolism, between the knockout and the wild type animals. And does this lead to any differences that we can observe in the lipid levels? So looking at cholesterol levels, total, free, and cholesterol ester levels, we do see an increase in the total cholesterol and free cholesterol within the wild type animals. And we see an increase in the total cholesterol in the knockout animals, but less so with the free cholesterol. This could be going into the cholesterol ester pool. But in both cases, in both genotypes, cholesterol levels are increasing in the liver. And when we look at triacylglycerol levels in the liver, we see a trend for an increase in the wild type, but a significant increase in triacylglycerol levels in the knockout animals with tunicamycin treatment compared to the wild type animals. So we haven't parsed out all the data to look at the changes in the gene expression to explain elevation in triacylglycerol levels versus the cholesterol levels between the two genotypes. And that's ongoing work, but it's giving is certainly promising to suggest that there's that STAR-D5 is driving some of these changes in the transcriptome that we observe after tunicamycin treatment. And so what is the model for STAR-D5 function in NASH? Again, I was suggesting earlier that STAR-D5 plays a role in moving cholesterol to the plasma membrane for efflux. And then under normal conditions, this is what would be happening. STAR-D4, though, now we have to consider this because we saw that it is ablated and it moves cholesterol from the plasma membrane to the ER. But if we have lipid metabolism dysregulation and with ER stress, what we're observing is that we lose STAR-D4. So this could potentially promote more accumulation of cholesterol in the plasma membrane that will then activate the signaling that I had talked about earlier that promotes proliferation, inflammation, and fibrosis through suppressing EGF receptor signaling and promoting toll-like receptor signaling. But if we have a loss of STAR-D5 after ER stress, we're still losing STAR-D4, that we can have in the absence of STAR-D5, perhaps we're having more of the retention in the ER that could suppress some of the signaling that we're observing up here through EGF and toll-like receptor signaling. And we see the diminished response that we're observing in our knockout animals. So what we have to look at next is, these pathways that lead to proliferation, inflammation, and fibrosis. But the challenge there is a lot of that's being driven through the hepatic stellate cells or the macrophages, which are minor components of the liver. So our next step is to do single-cell RNA-seq to try to parse out cell-specific responses in the STAR-D5 knockout mouse model to try to get a better handle on what's occurring. And due to the time, I'm going to just go right to the acknowledgements. We have, I know my grant has been scored. We had put in a grant to look at a high-fat diet PCB co-exposure model using the STAR-D5 knockout animals to see if that would impact NASH development. And I know the scores are in because the study section is meeting tomorrow to discuss it. So I will know soon how that will come out. But none of this would have been possible if it hadn't been for Dr. Kling, who's a longtime collaborator and friend and co-I on the NIEHS grant that we'll hear about, as well as on the recent funding that we received from the CIEHS as a voucher award to do the RNA-seq and a pilot project that is allowing us to continue the analysis of that data as well as look at PCB effects. And all the Bioinformatics was from the group Sheshra and Jenman Pan, as well as Eric Rushka and Kalpani De Silva, who have taken all the peaks, gave us the list of gene analysis, and then is doing further analysis for us through the gene ontology, Metacore, volcano plots, all that good stuff you see in the bioinformatics papers, so that we can get this work published. But I'm very thankful for that. And Sabine Weigel, who was instrumental in teaching and helping us getting the libraries prepped. Kellyanne Peel, I have to she is a technician with Dr. Kling and I, and she has done, I would say, 90% of this work, and is just, it would not have been, is not possible without her help. Ron Gregg, chair of the biochemistry department, and Hoffman, they've been generated the mouse model for me and supported the, funded the generation of the mouse model, and they're keeping them alive, I would say feeding them. And also I want to recognize two of the Summer Research Scholars Program and Distinction Research Track students, Vishnu Patel, who's a M3 student this year, and Sydney Travers, a M2, who worked over the summers and generated a lot of the work, and they are taking off and asking different questions because not only did I collect liver, I collected kidney and ileum and other tissues, so we can ask some questions in the future. So with that, I thank you for your attention, and I will take any questions that you may have. And here's today's event code. Oh, I see there's 2 chats. I don't. Okay. Does the total blood cholesterol correlate with liver cholesterol? Do statins lower liver cholesterol? Do statins increase the risk of diabetes? If yes, how would that happen? Okay. Well, the idea with the statins is that to lower liver cholesterol so that the body will take up more of the circulating cholesterol from LDL, which is the problem with this high blood cholesterol. So it should be, it should be an inverse relationship. So the higher your liver cholesterol, or I should say, would be parallel in a disease state, that you could project that a higher liver cholesterol and a higher blood cholesterol would coexist, when in a healthy person that shouldn't exist. Is that correct? Yeah. Statins decrease the biosynthesis of cholesterol in all cell types, but mainly in the liver, and that decrease will then allow for increase of the LDL receptor to uptake more of the circulating cholesterol. Do statins increase the risk of diabetes? Well, I know there are off-target effects of statins, but I can't address that. I'm not familiar with that. This group probably has.
Yeah, they do increase.
Okay. Okay, so I don't know if you heard that statins do have a small risk for increasing in diabetes, but the benefits of the statins far outweigh that risk, so they're still used. So I guess the mechanism would be, is it an off-target or is it cholesterol mediated?
No.
Okay, so that might be not related to cholesterol itself. And okay, if it's, I don't know. Oh. And does expression of STAR-D5 change with insulin resistance or diabetes? Great question. Classically, STAR-D5 has been labeled as an ER stress response protein. So that's why, again, another reason looking at tunicamycin to induce ER stress. But what we saw in our animal models, and a lot of that work came from cell culture models, and what we're seeing in the animal models is we're getting very modest, if any, effect on STAR-D5 expression, but we're getting a dramatic effect on STAR-D4, which is the other cholesterol transporter, which is something I want to follow up on. But I'll have to go look in the databases. I've been looking at NAFLD or NASH and cirrhosis, and that's about, I haven't looked at insulin resistance or diabetes. I'll have to go. Yeah, thank you, Steve. Okay, so tunicamycin was originally used to block glycosylation membrane proteins. How is that impact separated? Well, that's, yeah, that is a good criticism because this is a chemically induced ER stress response because we're mucking up the ER and you have that accumulation of those proteins because they can't get glycosylated and move to the Golgi. And so that is activating the IRE1, PERK, and ATF6. So how can that be separated? Well, we can use different, like fapsigargan, which is a calcium circa blocker, which also induces ER stress. I'm pretty sure that's it. So we could do differences, or they actually have other activators. I can look at that. So I can't, I can't say that it's the only thing that I could come back with is in the literature with tunicamycin treatment, looking at lipid metabolism dysregulation. All the responses seem to be consistent. Now, whether that's due to, and we can actually show in those cases, if you resolve ER stress induced by tunicamycin, that the lipid metabolism dysregulation resolves itself. So, but that doesn't directly answer your question. So, yes.
Is there a way to look at the subcellular STAR-D5? So, I'm even thinking, and I'm also IHC. Saw changes in retinol. Retinol esters are stored in stellate cells. They're increased abundance of stellates. It's only 4 days of stress in tunicamycin.
Yeah, so that's what I think with the RNA-seq, we're going to be able to tell a lot more what's happening in these different cell types. And I did IHC analysis on kidney cell lines at one time after tunicamycin, and we did see redistribution of STAR. But that was in a cell line. And also in that case, when I treated that cell line with tunicamycin, we got an induction of STAR-D5. Oh, and Steve, coming back to your question, in diabetic mouse models, we see elevated STAR-D5, but that's type 1 diabetic mouse model. We see an increase in STAR-D5 in the kidney. I haven't looked in the liver in that mouse model.
So how do you look in that model that was the STAR-D or so the first hit is still going to be triglycerides and fatty acids.
Right. So I guess I'm trying to make the argument that the loss of STAR-D5 would be a second hit, or STAR-D5 is a second hit. Can we target STAR-D5 to get rid? If we can diminish STAR-D5's trafficking, will we have improvement or slowing of the progression of the disease?
So there is one from going from fat accumulation, fibrosis.
Right, and I'm relatively new to thinking about this, but my impression now and my interest is looking at the stellate cells because it's real. How do they get activated? And if there's such a minor cell type that I might be missing what's really going on, or really where STAR-D5 is playing an important role, might not be in the hepatocyte, which is what we're looking at. We're looking at the changes in the hepatocyte, but it might really be either the macrophages, Kupffer cells, or the stellate cells. And I haven't been able to read on the epithelial, the sinus, yes, that have an important role also in liver disease. So I don't know about that. But it is, because we see a difference. I mean, these questions are great because it seems like how can one cholesterol transporter, do that? Maybe it's not related to cholesterol transport. Maybe STAR-D5 has another function that we don't know yet and haven't uncovered. I'm building my models around what is known, and reviewers want me to link it back to cholesterol trafficking. So that's what it is. So because it would be important then to see, is plasma membrane cholesterol levels different in the knockout model after you get rid of STAR-D5? To me, that would be a great way to test that potential model.
It's like what Dr. Venters on STAR-D5 and diabetes. Those seem to be much develop the fibrosis.
Right. I mean, it could be. Yeah. I mean, it's yeah. Right. So these are great comments and things to think about. But I will go back and look at that. I'll let you know, Steve, if I find anything. Dr. Winters is on the. Okay. Thank you.